Okay, this is the next video in the trilogy of action potentials. Have a look at what the refractory period is. Try to explain what we mean by the all or nothing principle and how altering the strength of a stimulus affects the transmission of action potentials. And then some other factors, so temperature, axon diameter, myelination, how they affect the speed of transmission. So, action potentials are transmitted along neurons as waves of depolarization. Meaning an action potential occurs in one place, that stimulates the action potential in the next region in the membrane, and then the next, and then the next, along the whole length of the axon. Okay. An increase in the permeability in one area to sodium ions then stimulates the voltage-gated sodium ion channels in the adjoining region to open, and so on and so on and so on. So it passes on the depolarization, much like the wave. Feel free to practice this at home. Okay, so this diagram demonstrates it nicely. When one region becomes depolarized, so voltage-gated sodium ion channels have opened, sodium ions diffuse in. As they diffuse in, they will begin to diffuse down the axon to the adjoining region. Those voltage-gated, or referred to here as voltage-sensitive, channels in the adjoining region are then stimulated to open. As they are stimulated to open, that then becomes the depolarized region. So sodium ions diffuses in, sodium ions diffuse down the axon to the next adjoining region, and so on and so on and so on, until, and this is a really useful phrase, the depolarization occurs along the whole length of the axon. Okay, this whole length of the axon is really important. It will continue until it reaches the end. Using that term along the whole length helps to explain some of the factors that affect the rate, the speed that it is transmitted. Okay, another term here to introduce, which is the refractory period. So the refractory period is the time taken for the redistribution of ions. So as soon as depolarization begins and until hyperpolarization finishes, this can be considered the refractory period. During that time, it's not possible for a neuron to transmit another impulse. Another action potential cannot be produced at that particular point on the axon during this refractory period. I mentioned in a previous video that this means that each action potential is discrete or separate. So only one action potential can occur at one particular place at one time. Therefore, there is a maximum frequency of impulses. The maximum frequency would be action potential, and immediately after hyperpolarization and the redistribution of those ions, immediately on the resting potential being re-established, another depolarization, repolarization, hyperpolarization begins. Okay. There is an important principle with action potentials. That is called the all or nothing principle. An action potential will always begin at the resting potential, become less negative, and reach plus 40 millivolts with depolarization, and then followed by repolarization until the resting potential is established again. The amplitude for any action potential is always the same. So a large stimulus will not produce a larger amplitude, but instead more frequent action potentials. And we call this the all or nothing principle. Okay. The second feature here is that the threshold must be reached. So if there is a stimulus, the stimulus allows some of those voltage gated sodium ion channels to open. When those sodium ion channels open, sodium diffuses in and the membrane potential becomes less negative. If it reaches this threshold potential, an action potential will follow. If too few sodium ion channels open and not enough sodium ions diffuse into the axon, the threshold potential will not be reached, and the membrane potential will return to resting. There will be no transmission of the action potential along the membrane. So we call this sort of subthreshold, below the threshold potential. So the principle here really is there must be enough sodium ions diffusing into the axon to reach this threshold potential. And that's only possible if enough voltage-gated sodium ion channels open. That's only possible if the stimulus is large enough. If the stimulus is too small, there won't be enough sodium ions diffusing in. The threshold will not be passed, meaning an action potential will not occur. So all means the threshold is reached, we get a complete action potential. Nothing, no threshold, no action potential, no transmission. So there's no such thing as a big or a small action potential. They all have exactly the same amplitude or height. How is a big stimulus coded for, therefore, then? The frequency. Okay, so a large stimulus will produce a greater frequency of action potentials which are transmitted along the membrane. And this graph demonstrates this. You've got this graph is in your notes, I think on page 18. So what we're looking at here is a stimulus being applied to one part of the membrane, and then the action potentials, or the membrane potential, being measured at another point. And often exam questions will do this. So if we think about our motor neuron, let's imagine that these stimuli are being applied at point A. Okay. And we've got three different stimuli. A small one, a medium one, and a large one. The membrane potential will be measured at point B. So that could be anywhere, sort of further along the neuron. Remember, the impulse will travel from the cell body and the dendrites all the way to the axon terminals. So with this first stimulus, the stimulus is applied. It is a small one. Some sodium ion channels open, some sodium ions diffuse in, but not enough to reach this threshold level. So, the stimulus at point A is sub-threshold. The measurement taken at point B, well, there is no change to the membrane potential. Second stimulus. This time is a bit larger. More sodium ion channels open, more sodium ions diffuse in. It passes that threshold potential. Action potentials are transmitted along the length of the membrane until it reaches point B, where they are measured. Okay, and it has a relatively low frequency. I mean, the frequency is pretty arbitrary here, but relatively low frequency because the stimulus is a smaller one. This largest, stimulus number 3, largest stimulus, the most sodium ion channels open, and most sodium ions diffuse in. Remember, a large and a small stimulus are differentiated by their frequency. So you get a much higher frequency of action potentials at point B, but the amplitude is the same. It's got the same, can't spell, amplitude, but the frequency is much greater. So again, this diagram also demonstrates that all or nothing principle. Small, sub-threshold, not enough sodium ions diffuse in because there aren't enough sodium ion channels open. We've got this idea of nothing. Still can't spell. Over here, okay, even if it only just reaches that threshold or just passes that threshold, in both cases, all. And then the size of the stimulus will determine the frequency of those action potentials. Really common sort of questions these. Look for the size of the stimulus, but please refer to there being enough sodium ion channels opening and therefore enough sodium ions entering. That's where students often miss the detail. Okay? It's not enough it's not enough to say it reaches threshold, you need to say why. And that's because sodium ion channels are opening and sodium ions are diffusing in. Right, let's move on. Okay, what does this graph show? Same thing again. The intensity of the stimulus at the bottom. If this was, like we said earlier on, at point A, well, we can see here the threshold must be just above that, 1, 2, 3, the fourth stimulus. Because at point B, there are no action potentials. Each of these, they have greater stimuli, enough sodium ion channels open, enough sodium ions diffuse in to reach the threshold. We get that idea of all. What doesn't it show? Well, it doesn't distinguish between the size of the stimuli because it doesn't show the increase in frequency of action potentials. Right, so there are factors then that contribute to the speed of an impulse. First one is temperature. Higher temperature means a faster impulse, or faster transmission of those action potentials along the whole length of the neuron. Why? Well, kinetics. More kinetic energy, so the ions have more kinetic energy, therefore they will diffuse faster, and at higher temperatures there will be greater enzyme activity. For example, more ATP can be produced because the rate of respiration may be greater. More ATP can be provided to that sodium-potassium pump to re-establish the resting potentials. Of course, at very high temperatures, those transport proteins and the enzymes involved in respiration will become denatured. Hydrogen ionic bonds break, tertiary structure changes, active site no longer complementary to their substrates. So that will stop impulses from being transmitted. Next, the axon diameter. Now, we don't really need to explain why this is, although there is a question in a minute that gives you a little bit of a clue. The larger the axon, the faster the impulse. And we know this because giant squid with their massive axons, 110 millimeters in diameter, have really rapid transmission. Now, you don't need to be able to explain why to the same level of detail, but it's, I think, due to less resistance of those ions travelling down through the axon and stimulating the adjoining regions that we mentioned earlier. I'll come back to another question on that. Right, this is a really important one. Here we've got two different neurons. This top neuron, what is the difference between them? Well, this top one is non-myelinated. And the bottom one, we can see these are the Schwann cells. Schwann cells produce the myelination. That is an extension of their cell membranes. So it's like the phospholipid membranes surround and wrap around the axon. And that insulates this axon, meaning that the only exposed regions are at these nodes of Ranvier, the spaces between the Schwann cells. And because of that, it's only at these nodes of Ranvier where action potentials are produced. So if we imagine on this non-myelinated neuron, action potentials will be produced here. That would stimulate the adjoining region and the adjoining region, and so on and so on and so on. Action potentials would need to be produced along the whole length of the axon until it eventually reaches the synapse at the end, the axon terminus. In a myelinated neuron, the action potential is produced at one node. It then jumps to the next node, and the next one, and the next one, etc., until it reaches the end. So that means there are fewer action potentials that need to be produced. They jump from node to node, which is quicker. So rather than having to be produced along the whole length of the axon, they're only produced at the nodes. Okay, so neuron B will have the fastest transmission. The action potentials only occur at the nodes of Ranvier, and this is the terminology we need to use in the exam. The action potentials jump from node to node. This is called saltatory conduction. Saltatory, something like that. Meaning it jumps from one node to the next. Whereas, in neuron A, action potentials will have to occur along the whole length of the axon, which is going to take much longer. It also means these neurons require much more ATP because this resting potential needs to be established at every single point along the axon. There's much more likely to be the leakage of ions from non-myelinated neurons, which might mean a large stimulus at this end producing a large frequency at this end. By the time it reaches the other end, the frequency might be much lower. So you get this leakage of transmission. Okay, here's this idea of saltatory conduction. From one node to the next, sodium ions diffuse in at one node. They diffuse down through the axon, stimulating the voltage-gated sodium ion channels to open at the adjoining node. And again, it's much faster. Okay, saltare meaning to leap in Latin, so we call this saltatory conduction. So you've got this diagram in your notes. It shows the relative sizes of what we call these local circuits. So a much longer local circuit here means that a much the same distance can be covered in a shorter period of time. This is the myelinated. This is the non-myelinated. Okay, in your workbook, pages 20 to 23, I've got two questions, question 10 and question 11. I'm going to go through a couple parts of those, not all of it, but I'd like you to do that. So it's up to you. My advice would be to have a go at doing those questions now, pause the video or come back to the video later. I'm just going to take you through a couple of examples of sort of common themes that pop up. Okay, so hopefully you've paused the video, you've gone away, you've done it. If you haven't, do that now. Come back in a bit. This is from question 10. This shows a motor neuron. A motor neuron is the only neuron that you need to be familiar with, that you need to be able to label. A here, we can see the exposed region of the axon. B shows a Schwann cell. Now I want to be a little bit careful with this. It is pointing at a Schwann cell. The Schwann cell produces the myelin sheath. Now, I think the exam board would probably be generous, and if you'd called that the myelin sheath, you'd get away with it. But really, we are looking at the cell. So if you have a question like this in the exam, my advice would be to go for Schwann cell. And if you want to go belt and braces, you could say Schwann cell, that produces the myelin sheath, but I don't think that would be necessary. Lastly, there is that node of Ranvier. And you can see in this diagram that there are these spaces between the Schwann cells where the axon is exposed. Okay. 10.2 asks, explain how an increase in the temperature speeds up the conductance of a nerve impulse. And we've covered this already. So increased kinetic energy means a faster movement of the ions. Therefore, there will be a higher rate of diffusion of those sodium and potassium ions out of or into and out of the neuron. Now, this is a graph that I mentioned earlier on, sort of briefly, relating to the axon diameter. Now, I said earlier, you don't need to know exactly why a larger diameter will increase the speed of transmission. So on the x-axis, we've got the axon diameter in micrometers, speed of conductance in meters per second on the y-axis. Okay. Now here, you are asked to suggest. So you don't need to know this, but you can work it out. So as the diameter of the axon increases, the length of myelination between the nodes increases. Okay, so bigger diameter, the Schwann cells increase in length, there's more myelination. And therefore, the distance for these jumps between node to node, so the action potential is produced only at the nodes, or depolarization is only at the nodes. The depolarizations jump from node to node. Well, if the myelination increases in length, it's going to jump further. and therefore the impulse will be transmitted further along the, or faster along the neuron. Okay, so any question about saltatory conduction? To begin with, impulse jumps from node to node. Sounds really straightforward. That's fine. I'd probably go for depolarization only at the nodes, but you'd get away with that. If the myelination increases in length, there will be fewer depolarizations required. And here's that phrase again, to travel the whole length of the axon. So it's about getting from one end to the other. Okay, so that's all I was going to go through, because those two questions are pretty typical for transmission of action potentials. But there was one last little bit that I'd like to introduce, which is just about it's sort of an application of this principle. And that is some damage done to the myelin sheaths with certain conditions. Okay, so if the myelin sheath becomes damaged, the rate of transmission along that myelin sheath will slow. Why is that? Now, you're not expected to know about each different condition, but you could work it out. Rather than impulses only occurring at the nodes and jumping from node to node, as it would do in a healthy myelinated neuron, when the myelin sheath becomes damaged, you jump from node to node, but then we've got this idea of a non-myelinated bit. Okay, so impulses are produced over a greater surface area or over a greater length along the axon before it can reach the end. Okay, so we see this in multiple sclerosis, where the myelin sheath can be damaged, plaques build up on the myelin sheath, which damages the myelin sheath, meaning the speed of transmission becomes slower. Okay, there is a greater surface area where action potentials must be produced. Those depolarizations are occurring along a greater length, and therefore the speed of transmission slows down. There is also a bit of an issue with multiple sclerosis, with the jumping of impulses from one axon to a parallel one. Okay, so we can get leakage, and sometimes that means that if we imagine that these are two parallel neurons within a nerve fibre. So a nerve fibre is a tissue, is a bundle of different neurons. Each of these neurons, these motor neurons, is going to end at a different part of a muscle. Okay, and to ensure real fine motor control, impulses are sent along very specific neurons to stimulate specific muscles. If there is leaking and impulses, action potentials passing from one neuron to another, perhaps a different muscle than the one intended will be stimulated. And that can cause, imagine, let's say that there's another neuron with a neuromuscular junction over here. Rather than this region of the muscle being stimulated and contracting, a different region contracts, and then we get this loss of fine motor control, or in some cases tremors that you might that you might be aware of as one of the symptoms in with certain diseases related to damage to the myelin sheath. Okay, right, I'm going to stop there. I hope you found those 3 videos useful. I will come back with another one on synapses next week. Okay, cheers.